Oh my gosh. I'm just gonna make a poll real quick. They don't have any like Kahoot today, but that's what I use for my attendance. So I'm just gonna make a Kahoot really quick and just use that. This will be. Yay.
Those, can you have a Quizlet premium account I could possibly steal from you?
A what?
A Quizlet premium account?
I don't use Quizlet. I have Kahoot.
I've
So-
been using Quizlet to make like study guide Quizlets for
Yeah.
everybody,
Uh-huh.
but kind of sucks if you don't have premium, and I'm kind of poor.
Yeah.
I can't afford premium. So I texted my friend Fish, and I was like, hey Fish, you got Quizlet premium? He's like, no, that sounds like something I have. Grab Kahoot premium, I don't need Kahoot premium. It's fine, it's really fine.
Yeah, I mean, yeah, because you can only make like a certain number for free or something like
You
that.
can
right
only
now.
make a certain number of questions, study questions for free. And you can only do a certain amount of study questions for free.
Yeah.
Which is very aggravating. I'm still waiting for my birth card stuff to come in, because Oklahoma promised her. Whole thing.
Yeah. But I don't have to go that well. Say that. All right. No, we're doing calling on it today.
But physio, yeah, but I passed it. Like 88.
All right, let's get signed in. We're gonna get going with this. Chapter 6, if you haven't looked in at all at chapter 6, try to prep for it. It's a heavy chapter, y'all. So we need to get cranking with it. Pretty much all of the chapters that we're doing up until maybe the last two for this. 13 and 14 aren't as bad. 13 is not that bad. 14 is mostly just terms. But the others, 6, 8, 9, and 25, and then 10, those are heavy chapters. So get ready for a lot of info flying at you again. Like I said, this class pretty much doesn't stop with that. So like unit 4 is not that bad because we're using a lot of that information and just applying it to diseases. Anybody have any questions before we get started with the Kahoot? Any questions about the test? I still have people that are waiting to take the test that have had the flu. And so I will have that opened up once that is done. That should happen sometime this week, assuming that the weather is going to accommodate, right? So that might delay that as well. But yeah, we had a question about what will happen if we do have weather. If we have to close for snow or ice day or whatever, you will be responsible for learning whatever chapter we would have learned that day. So there will be videos available for you guys that you guys will be required to learn. And then there will be a Kahoot that you guys will be assigned that we would have done that day. Yes.
Well, it's like,
I forgot about that part of it. That was my fault for that. I don't want to have to change it now, but I'm sure I'll be able to figure it out, right? You guys are mostly using your names, right? Did you use your first name for it?
yeah.
We should be good then. Yeah. As long as you guys are using like your name or some version of your name. I forgot to add the email part. That was my fault.
I just made.
Yes. The lab. So if we have an ice day, we will still have the lab. We'll just combine it with another lab day. So that does mean that there will probably be a lab day coming up that's going to be crammed with 2 labs. So be aware of that. We'll consolidate as much as we can. Luckily, labs 8, 9, and 10, they're shorter days. So it should accommodate the one that we're doing on that day. And as long as you guys have been working on, because the lab I think we're going to be missing, of course, is not going to be today's lab because we'll get that one in. So I'm worried about us missing lab 6, and that's our motility lab. And that requires really like the only constraint, the only reason that one takes, it's not usually long, but the only reason that it even takes time is finding your organisms in the microscope. So as long as we're good with our microscopes, that lab shouldn't take too much tacking it on to another lab. So yeah, I'll say that is like, the only thing with that lab is the motility lab. We don't stain our cells. So it's finding unstained cells. So they are very hard to see. So that's the only issue with that one that can sometimes add on some time. But the other labs that we have, lab 8, 9, and 10, lab 11 we usually take most of the day, but those 8, 9, and 10, it has some leeway in there that we should have given attack another microscope. Not too worried about it. But yeah, so I won't have it be where like if we have a snow day that you guys have to join in on Zoom at 10 a.m. That's what I'm saying, right? So there won't be a requirement for you to do something that day, but you will still have to be sure you learn that info. Does that make sense, everybody? Okay. Y'all, so we went to the grocery store on Super Bowl Sunday, which was a bad decision. Right. Well, we were just like, we just wanted some Coke Zero because we ran out. My husband drinks a lot of Coke Zero. Right. So it's like at that point, we didn't realize it until we pulled into the parking lot. And I'm like, my God, we're already here. I'm going to sit out here in my car and wait on you while you go in there and deal with this. So he went in there to go get Coke Zero and some other things. And it was a nightmare. And it wasn't just because of Super Bowl Sunday, it's because everybody's trying to go to the grocery store in Armageddon, you know, and usually I like to go in there and make a big show of it and make a big scene and be like, oh my God, Snowmageddon, I'll get like armfuls of like rice-a-roni or something and make a big scene because I like to be that person. But I didn't want to go in. There was too many people in there. Like it was like lines to the self-checkout, like all the way to the back of the store. Yeah, and that was in Yukon. I don't know where I was, where you guys were. We do our grocery shopping in Yukon.
These were busy plans. I stayed 3 hours past my schedule time.
Yeah, I bet even in Sprouts, like that's something, like,
I worked at Subway in high school and I didn't go home until like 1 AM
yeah.
on some full Sunday.
All right, we ready, guys? Let's go ahead and get going. It's just one question, it's just a poll. But it's just for me to get the attendance knocked out. What do you think's gonna happen with the weather? So there's only 4 choices, and if you guys pay attention earlier, you saw me type in the choices.
I already paid for it. I already paid for my active place.
Yeah, I think that that is on this top one, this one here, the red one. I think that's most likely. I think we're gonna close early. Like, that'll be open most of the day and then like close the campus at like 4. Cause I think it's gonna start dropping temperatures around then, start raining and
The ice rain.
I have a late class, so I'm actually worried about that on Tuesday. So, but yeah, that's what I think. All right. Yay, we did it. How do I finish this? All right. Anyways, moving on. Let's do it. All right, so before we get started, our questions for the end of the chapter, because we're back onto that again. Our questions at the end of the chapter, I'll take care of the first one. Then we have Gavin for question 2, Gina for question 3, Jessica for question 4, and then Reagan for question 5. If I called you out and you were on Zoom, you are still responsible for your question. All right, so let's talk about genetics. The genome is all of the genetic material in an organism. So every last bit of genetic information in an organism, including any extra chromosomal genetic information. So that means things that are outside of the chromosome. All of that is the genome. So most of that's in your chromosomes, but like when we talk about our mitochondria, they have their own DNA. So that would be included in our genome as well, okay? So things that are also not chromosomal would include plasmids. We talked about these guys with the bacteria, right? Those are little extra bits of DNA that are circular. So extra bits of DNA that are circular that usually has genes that help the bacteria survive. And any DNA that's in our organelles, like we said, the mitochondria, the chloroplasts also have that. So the genomes of your cells, if you are a cell, your genome is DNA, period. So if you're bacteria, archaea, or a eukaryote, you have DNA. But we already learned that the viruses can be DNA or RNA, and they can be single-stranded or double-stranded. And if they're single-stranded, they can be positive sense or negative sense. And that's going to make more sense after today's lecture, okay? A chromosome is basically a physical structure of a package of DNA. So we have multiple chromosomes. We have 23 pairs of chromosomes, so 46 chromosomes altogether. 1 each from the paternal side and 1 each from the maternal side. And then, so that's basically how eukaryotic chromosomes will be going. We usually occur in pairs for most of the time. But our gametes will be haploid, so they'll be in singles. So only have 1 representation of each. So your eggs or your sperm only have within those cells 23 chromosomes, period. They don't have pairs in them. All right, and ours in eukaryotes are always linear. Whereas bacterial, single circular chromosome. And it is double-stranded, just a helix, like a double-stranded helix, but it's just 1 big old circle. All right, a gene is a informational packet in which we would divide our chromosomes up, basically. And we have different categories of genes. The structural ones, structural genes are the genes that code for proteins. Then we have genes that code for RNA machinery, like our ribosomes, or like those are made up of ribosomal RNA, or our transfer RNAs, which we're going to talk about today. And then we also have regulatory genes that control the gene expression. So basically your genes are only going to code for proteins and then the things that either build or control making those proteins. So that's it. All right, there's 2 terms relating to your genetics. There's real basic genetics that we're talking about today. The genotype and the phenotype. Your genotype is your entire genetic makeup. This could include the fact that, like, I have green eyes, for instance. I might have a recessive gene on like my dad's side for like blue eyes, but I'm expressing green, right? So you would have genes for green and blue, but I'm only expressing one of those traits. So that fact that I could be carrying genes that I'm not expressing, that's contained in my genotype, but your phenotype is what you see expressed. So you can be carrying genes that aren't being expressed. Okay, those are called recessive genes. We've learned about that. You can be carriers of those genes. A lot of times you need 2 copies of recessive genes in order for them to be expressed. So if I don't know if blue eyes is recessive, but if blue eyes is recessive, then you need 2 blue eye genes in order to have blue eyes. But if blue eyes is recessive and green eyes is dominant, then you need 1 green eye will always trump the blue eye. And there's a lot of in-betweens in genetics and stuff like that. But the basics is you'll have dominant genes and you'll have recessive genes. You only need 1 copy of a dominant gene for it to be expressed, but you need 2 copies of recessive genes for them to be expressed. What happens if you have 2 dominant genes? It's just dominant. They're just dominant. So we're going to talk about in chapter 17, blood groups. In blood groups, when we talk about A, B, O blood types, okay, A and B are both dominant traits. So if you have a trait for A from one parent and a trait for B from another parent, you can have AB. They're both being expressed. Okay. But if you have A from one parent and O from another parent, O is recessive. So you'll express A, right? If you have 2 O's, then you'll get an O. Interestingly enough, anybody know what the most common blood type is?
Yeah.
It's O positive. Yeah, it's O positive. So there are plenty of examples where you'll actually see, even though it's recessive, doesn't mean it's the least common. That usually doesn't have really anything to do with it. But O's can be carried around with the other traits, right? And so you actually see a lot of O's coming together and making O. So O positive is the most common of the blood types. AB negative, I think, is the least common. I think that's the least common. My sister's AB positive. If you guys know anything about blood types, again, we'll talk about that in chapter 17, but AB positive is the universal recipient. So if you have AB positive, you can receive literally any blood type at all. But if you're O negative, you're the universal donor. Anybody can receive O negative, but O negatives can only receive O negative. They can't receive any other blood type. So we'll talk about that in chapter 17. But that gets us to talking about genotype versus phenotype, doesn't it? So now we can start seeing a very good example of something that a lot of us are familiar with so far, knowing the different types of blood types, that your genotype and your phenotype can be different, right? So you might have 1 gene for A and 1 gene for O. That's your genotype, but your phenotype would just be A. That would be your blood type A. We get that? Okay. Phenotype is what's expressed. All right, the structure of DNA. DNA is, deoxyribonucleic acid, and our little monomer subunits that are going to build our DNA. Those subunits are called nucleotides. And each nucleotide has parts. Those parts are phosphate, deoxyribose sugar, and the nitrogenous base. All right, so our nitrogenous bases determine basically what your DNA is spelling out. And these are your A, T, C's, and G's. Now remember, I had told you guys previously that the way that I remember these, move this out of the way, is basically like when you look at A, always binds with T for DNA. To me, A and T, they have harsh lines, right? Whereas G always binds with C. They both got that curve going on, C and G. So these guys are always going to be making hydrogen bonds across with one another in that double helix. A never binds with G or C. It only ever binds with T across the way. So now if I were to explain to you that one side of my double helix reads A, C, C, G, T, you guys would be able to tell me what the other side says, right? So A would bind with T, and then G, G, C, A, right? So now we know we could actually like write out our whole DNA on both sides if we needed to. So that's what's going to be making up our double-stranded helix, okay? Our double-stranded helix is arranged in an anti-parallel arrangement, okay? So now we're getting down to the chemistry sides of things. 5' to 3' in one direction, and then the other side is going to be going the opposite direction. So what the heck does that mean? Well, I'm going to show you what it means. So this is basically how we're going to be looking at this, all right? And you guys are going to be hearing me say everything runs in 5' to 3'. Everything's going in 5' to 3'. This isn't something that's coming up and you get to forget about it and not bring it up because this whole chapter and the next chapter pretty much. I'm gonna be like, so remember that this synthesis is happening in the 5' to 3' direction. All the synthesis that we do, DNA or RNA, when we're making copies of things, always runs in the 5' to 3' direction. So this is going to come up. I'm going to show you what this means in this thing here. Basically, we have numbered our carbons in our fancy little sugars, okay? So remember how a nucleotide is going to be your phosphate with a sugar, a 5-carbon sugar of some kind, right? And we know that that looks like a nice, happy little pentagon. And then that's basically attached to a nitrogenous base of some kind, and that's got a nice little structure to it as well. All right, so down here, we have a phosphate, we have a sugar, and we have a base. So this thing right here is a nucleotide, okay? And it's just repeating nucleotide all over and over and over again. So what does 5' and 3' mean? Let's erase this. So on our sugar, we can actually number these carbons that are on here like 1 through 5, okay? And they have done so. So we have basically 1, 2, 3, there's a carbon here, and then we're going up to here. Okay, this is the linkage here. So this is how they're numbering them. Don't worry about that because I'm not going to ask you to pick it out or tell me which one is the 5' carbon or no. There's never going to be any of that. Well, I just want you to understand this isn't just pulled out of our butts out of something. This is going to tell you what is actually making up that orientation. So what you'll see is, you see how this phosphate up here goes to this branch and then down to the sugar? But you look on the other side, what do we see? The branch and then the sugar and then a phosphate, right? So you see how these are oriented in opposite directions? That's what this is talking about. That's anti-parallel. So here we're going from this end to this direction, whereas the other one is literally arranged in the opposite fashion. And what am I talking about specifically here? Those sugar phosphate backbone. So you can think of the whole backbone of all of DNA as basically being phosphate, sugar. And then off of that sugar, you have your base, okay? Now this is a nucleotide. I wish I could just copy that because that would make it so much better. But then we have another phosphate, sugar, and a base. Phosphate, sugar, and a base. This is basic, like how it will be set up, right? So if this is a nucleotide and this is a nucleotide, then the other side of it, this links to a base, this links to a sugar, but then this would be a phosphate down. You see what I'm talking about now? Okay, so they're oriented opposite directions. And that's based on the sugar phosphate backbone, right? That's anti-parallel. I won't show you pictures. I won't make you draw it for me, but understanding that anti-parallel aspect of this kind of dictates how we're going to be making our copies and how basically all of our functions of making DNA and RNA and all that drives everything that runs in your cell is basically dictated by that. So, all right. So that's the gist of that. What makes up something anti-parallel? So when we are replicating DNA, so DNA replication is just making copies of our DNA. If we are making RNA to do something, it'll get its own term, okay? If I'm talking about replication, I'm talking about making a copy of that molecule into an exact copy of it, all right? If we go from DNA to RNA, is that making an exact copy of that? No, because RNA is a different chemical structure, right? So that is not replication, right? So DNA replication is semi-conservative. And this picture likes to pretend like it knows what, that makes sense to you guys when you look at it. But basically, when you have a double helix and you have your ladder like this, right? And this is normal like DNA. Whenever we're making a copy, basically, you will take one old strand, which old will be red, okay? We'll separate our old strands. So this one will be the top old strand, and this will be the bottom old strand. All right. Then, bear with me. I'm sure you guys know where this is going. So we'd have our ladders that we would bring to make new strands. So whenever we're making copies of our DNA, it's part old and part new, always. So if you're going from making a copy of your cell, you skinned your knee and now you need new cells to fill up and make new skin cells to cover up that wound. Or you're growing as a baby and you need to duplicate your cells or whatever. Every time you're copying your DNA, that new copy that you've made, the two cells that you have as a result will have half old and half new DNA, half old and half new DNA. It's not like you have old DNA and new DNA in that cell, okay? Each of the new daughter cells consists of old DNA with new DNA. And we use the old as the template to create the new. That's called semi-conservative because we conserve half of our DNA. So semi-conservative, right? So this would be in one cell and this would be in a whole other cell in our daughter cells while we're going through mitosis. So think about that in the big picture of that, okay? So in this case, they use the opposite colors. I should have paid more attention to that, but these opposite colors where our blue is our old, okay? And we're opening it up. And then our DNA polymerase, that's going to be the polymerase polymer, making copies of repeated units of our monomers, and that's our nucleotides. So DNA polymerase, and ACE means enzyme, is the enzyme that's going to be making copies of our DNA. And as that's happening, we're going to be opening up our helix. So it's like closed, it's opening up, and we're reading one side and making a copy of it, and reading the other side and making a copy of it. Our copy is always made in the 5' to 3' direction. We can only add on nucleotides in the 5' to 3' direction. All right. So that creates a problem. So semi-conservative, each daughter molecule is identical to the parent in composition. But neither one is completely new and neither one is completely old. Half and half, right? So it's semi-conservative. So that enzyme, DNA polymerase, you don't need to know that it's DNA polymerase 3, just worry that's DNA polymerase, okay? Once we unwind our strand and have it out, it's just a flat old ladder because we have to unwind it first. We'll separate them. And then our DNA polymerase will synthesize our new daughter strand using the old one as the template, okay? We can only add to the existing chain. So we can't just start off of nothing. So what that means is once I separate my chains and we have our happy little fork like it was showing us. So your helix is going on down here. And now we've basically opened it up to where we have this closed. And now we're opening it up onto the two sides, right? And we're going to make copies. So we're opening it up and we want to start making copies. You can't just start adding nucleotides out of the freaking blue. So I say we will synthesize in the 5' to 3' direction. Well, if this was the 5' and then we're going into this way would be the 3'. You can't just start adding to nothing. So we'll have to add on to something. And so that usually means we'll use RNA as a primer and then build off of that and then go back and replace that RNA later on, okay? So we'll talk about primers a lot, especially in terms of genetic engineering later on. But it's essentially just a temporary bit of DNA that we're just using to build off of. We can't build off of nothing. So we can only add, that's what I'm talking about with this. You can only add nucleotides to an existing chain. Okay. And we can only add nucleotides in the 5' to 3' direction. There's rules we got to follow with this. So this is going to create a problem and you're going to see it here in a minute. All right. So as we're opening up our helix, because we can't just open up our entire chromosome. I mean, we're talking about billions of nucleotides. We can't just open it all up all at once. So that place where we're opening it up, that point where it comes to that point in this image here, this point right here, is called the replication fork. Okay, that's this. And then that primer is a little bit of RNA that's single-stranded that we use just to add on new nucleotides for our DNA, and we'll replace it later on. So we would add a primer here. And then this would build in this direction. And then you'd imagine that this other side would be building in this direction. Because again, if it's anti-parallel, we're going to be building in opposite directions. So this would be our 5 over here. This would be our 3. But now you're starting to see if we started pushing this fork open in this direction, you can just use your little happy polymerase and just read on through, right? But if your polymerase can only add in a 5' to 3' direction, uh-oh. Now, as you open this further, how are you going to get these guys taken care of for this, right? This is going to create a problem because you can only create so much. You have to, as you open it, this side up here at the top can just read through smoothly. You just push it open. We're just going to keep on reading through one big old nice long strand. But the other side, as we open up, we're revealing more space that we needed to fill. And we didn't have that open earlier. And as it's getting pushed open by the other side, we'll have to make it in fragments, okay? And that's going to show that. So the leading strand is the strand that will go continuously in that 5' to 3' direction. You'll just read on through. That's the leading strand. This is leading the whole process, all right? The lagging strand is the strand that we have to make in short segments. So one side gets made continuously, while the other one, as we open it up, we'll fill in those gaps as we go in parts, okay? And those segments are called Okazaki fragments. Okay? Those segments that get made, they're called Okazaki fragments. And it's just because that lagging strand has to be made discontinuously. We have to make it in little fragments. As we open it up, reveal some nucleotides that need we match up, and we'll just go in fragments as we open it up. All right, so this is the idea. In your 5' end, we're able to open up this fork and just keep on reading through. And as we open it up, you see how now we've opened up some nucleotides that we are going to need to make. We can only go in our 5' to 3' direction, which is this way. As we open it up, we'll make our little fragments here, and that's what these guys are here. That's an Okazaki fragment. Okay. All right. Fragments can't just exist as fragments in your DNA. Your body can't deal with that. It doesn't know what it's supposed to do with fragments. So we seal our fragments closed. Our Okazaki fragments. Hold on. Oh, dang it. Using an enzyme that we call DNA ligase. To ligate something means to connect it. Anybody in here ever play a woodwind instrument like a clarinet or saxophone or something? Okay, so I play tenor sax. And when you put your reed on your instrument, you use a ligature to hold it on, right? And so ligature is holding the thing together. So ligate, that's how I remembered it. That really only helps a select few of you. But ligate means to join together, to bring it together, hold it together. So DNA ligase, like it shows here, this is our green guy here, is going to seal together your Okazaki fragment so it's all one continuous fragment. So your body can actually use your DNA the way it needs to, okay? So there's a video.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
All right, so that is the video version of what I try to draw for you guys, to try to make it a little clearer for you guys so we can see it in practice. So are we starting to understand now what a lagging strand is, what versus the leading strand? Leading strand just goes straight through. The lagging strand is made in fragments as we open up that fork because of the way that the DNA polymerase has to only go in one
Here is the
direction.
cell,
Sorry.
the basic unit of all living
There's a
tissue.
cell, guys.
So,
All right. Escape. I'm gonna go back to my slide. There we go. All right. Because we're gonna move on. We're done with DNA replication. That's all you're getting for DNA replication. All right. And yes, I could easily cover that for days, but we're gonna just, that's it. All right. So we're gonna move on to what the point even is of DNA. Now we know we need to, if we're making new cells, each of our new cells has to have its own DNA because everything that cell's gonna do is coded by your DNA. But how does that work? So that's gonna bring us to our central dogma. The central dogma of genetics says that DNA is used as our basically blueprint housed in our nucleus, or if you're bacteria, it's just hanging out. Just saying, don't forget that. And that we're going to turn it into RNA via a process called transcription. And that RNA is going to be read by a ribosome using things called tRNA. Together, they're going to work together in a process called translation to make protein. Okay, so this is the central dogma of genetics. DNA begets RNA begets protein. And we used to think it only went in that direction, but we've already talked about how some viruses can go from RNA back into DNA. And that was like game changing. Okay, so transcription and translation. Very important not to confuse the three topics that we're introducing here. Replication, making a copy of DNA. Transcription, going from DNA to RNA. And then translation, going from RNA to protein. So replication, making the copy of the original. It's just a direct, exact copy. Okay. Transcription, we're going from one kind of nucleic acid, so deoxyribonucleic acid, to just ribonucleic acid, right? But are they still nucleic acids? Yes. So think of that as writing down a noted version of how to build something, right? You're transcribing. If you're a scribe, you're writing. It's still in the same language. You're just copying down what your instructions are just for that one gene. So you go to a gene, you make a working copy of just that gene, not the whole DNA, because we have 20,000 genes in your DNA. So we'll just go get the one gene that we need to build a chair. Okay, so you go to the Smithsonian Institution, you copy down instructions for how to build a chair, then you leave with that copy to go home to build your chair. You don't build it in the Smithsonian Institution, right? You go into the DNA in your nucleus, you make a copy of building chair, and then take that out into the endoplasmic reticulum outside of the nucleus, okay? That's transcription, writing down our working copy of that one gene. Okay, so that's what our messenger RNA is going to be. Then we will take that messenger RNA and use a machine called a ribosome, which also happens to be made of ribosomal RNA, and using transfer RNA through a process we're going to go through in a minute, translate it And now we're taking it into a different language. Whereas before we went from nucleic acid to a different nucleic acid, but it's still nucleic acid language. Now we're going to translate it into a whole new language. And that new language is proteins built by amino acids. Okay? Different language, we're translating it. Okay? So we're going to talk about the processes here. Basically, your DNA and your traits are ultimately coded by your genes, and which gene we're going to be using and how just determines what your phenotype is, what you're expressing at any given time. And your DNA, where you have two strands, where one will be 5' to 3', and the other one will be this way. Okay, that's definitely a 5. You can so totally tell, okay? All right, so that we would have one strand be a coding strand and one strand would be a template strand. So you see how if we look at this strand here, this bottom strand is exactly pretty much identical to the mRNA strand, but we didn't use that to make, that's not the template that we use to make it, that's the coding strand. Yeah, it's going to be like this today, okay? This is the template strand up top. So where if our DNA said A, T, C, G, G, then our DNA strand and our other side of our DNA strand would be T, A, G, C, C, okay? You see how that would work, right? We can at least agree on this, right? That this is what your strand of DNA would look like. Then your mRNA would read U, A, G, C, C. Because RNA uses U, not T. But you see how this strand says the exact same thing as this strand? This is our coding strand. But we had to use this one as the template to make our mRNA. Used the template to make the mRNA. It just so happens that the other strand of DNA reads the same thing because the base pairing. Okay? And which strand is which depends on what gene you're looking at. Okay, so we can divide this down into 3 nucleotide sets. So if this was G, A, C, then this one would be C, T, G, right? I wish I'd used a different color. Let's do it. Blue. G, A, C, and this is C, T, G. Okay, can we agree on that? And then if this one was C, C, A, this one will be G, G, T, and whatever. And so if we were using this top one as our template, right, then this one would read C, U, G, or then this would be G, G, U. Okay, you see it? We getting there? Okay, that's how that would be divided up. And we divide these into 3. That set of 3 in DNA is called triplets. The set of 3 in RNA is called a codon. Okay, we just put NT here, right? The set of 3 nucleotides for our codon, for our RNA is called a codon. And it's actually going to be important that we follow these terms, okay? Triplets in DNA, codon in RNA. We're about to read based on those codons. Every 3 nucleotides, these codons here, this codon codes for 1 amino acid. All right? And that's how you go from DNA, transcribe it into RNA, and then we're going to go take that RNA and turn it into amino acids that are going to connect together to make a protein. So that's a big picture, okay? So these are major components of all this. Our messenger RNA, this is our working copy of a gene. Our transfer RNA, the reason it's called transfer RNA is because it's going to transfer amino acids. So this carries our amino acid based on what the codon pairing is. So this is how that would work. If your mRNA, so that's what this is down here, reads AUG, then it has an anti-codon. So see, now you need to know why a codon is a codon. So if your codon reads AUG, your transfer RNA has an anti-codon that matches up to it, A to U and G to C, like that. See how it's complementary? That when that matches up, it knows to attach to a certain amino acid. As it turns out, if your codon says AUG, the amino acid that's going to be attached up here will be methionine. What?
Start.
Yes. Yes, it means start, yes, but it actually, yes. So yes, it means start. So I'm assuming you guys are recalling it from another class that you took. That, yes. Okay, yes. So that's our start. AUG is always our start. And I'm not going to test you on that, but I'm glad that you remember it. I'm like, yes, it does mean start. Yeah, so you're always going to start with methionine. You're always going to start with AUG, as it turns out. And AUG is going to code for methionine, and that's always going to be your first amino acid on every protein. You might clip it off later, though. You might not keep it, but it always is starting out with that. And that is literally our sign to have our ribosome attached here and start here. And everything after that is going to be read based on our codons and what your codons are going to call for. No. So stop codons are stop codons. But your anticodon is literally on the tRNA. So this is what this whole thing is. This whole weird T-shaped thing is a tRNA. Okay. So the anticodon is the complementary sequence to the codon on the mRNA. And if your tRNA anticodon matches up with your codon on your mRNA, then that means that we will attach this amino acid. Okay. So if your codon reads CUG, then your anticodon would be GAC on your next tRNA, and then whatever amino acid that codes for would be attached onto that. Okay, you seeing it a little bit? This is transcription and translation. This part right down here is called translation. We're going to start marrying these processes together. Okay, so making our messenger RNA, that's transcription, and translation is using tRNAs and ribosomal RNAs to make the protein as a result. So we're going to show a video of that too. So we're going to get there, but we're explaining our purposes of our things. Ribosomes are made up of ribosomal or regulatory RNA. And then we have enzymes that might be involved, and we have a whole lot of raw materials that are going to be involved and whatever. But you better know the difference between, and we'll always call it ribosomal RNA in this class, okay? Ribosomal RNA makes up the ribosome. Transfer RNA carries your amino acid that'll match according to your codon on your messenger RNA, which your messenger RNA is your working copy of your gene. All right, we know RNA, we usually are going to see it as a single strand. It's going to contain uracil, and our sugar is ribose. It's not deoxyribose. Okay. Messenger RNA, working copy of a gene. Okay, so it's a transcript. That's what it's called because we transcribed it from the DNA. It's a transcript of a gene in the DNA. Is made in a very similar process as how we do DNA replication, except we're using RNA instead of DNA, right? So it's a different chemical structure. So this is transcription. And those codons that we talked about, triplet of bases, so 3 bases, and that will eventually code for 1 amino acid. That's the whole point of them. So literally your entire DNA is literally all your A, T, C's, and G's. You could divide it into 3s. Those 3 triplets in your DNA are going to equate to a codon in your messenger RNA, and that codon is going to make 1 amino acid. And that's how your DNA becomes protein. All right, your transfer RNA, this guy is going to take complementary sections that are going to match your tRNA to your mRNA codon. And those complementary sequences are called anticodons. And that when that matches, that's going to lead to the appropriate amino acid being added on. All right, so that's what that anticodon is. At the bottom of your clover leaf of your tRNA, this is your tRNA. This is where your anticodon would be. That matches up with your mRNA codon. And this is all going to happen in the structure of a ribosome. All right, we're starting to see it a little bit. There's videos, don't worry. All right, so this is a fancier picture than what I can draw of it. This is our ribosome, and this is where all the magic is going to happen. Where our little tRNA, which is this guy here, will come in and match your codon on your messenger RNA transcript and read along the whole thing, just adding in amino acids and linking amino acids together basically as we go. That's going to be translation. So first things first, how we go from DNA to make our messenger RNA transcript. Okay, this is called transcription. So DNA to our messenger RNA, called transcription. The first step is initiation. Easy, these steps, okay? Second step is going to be called elongation. And the third step is going to be called termination. Easy enough? So initiation. Initiation is when RNA polymerase binds at a spot that we call a promoter, which is basically where RNA polymerase starts, okay? Like the start point for RNA polymerase is our promoter. And once we bind there, we'll be able to have our unwound helix and use one strand as our template strand to build our transcript off of. So this is what it will look like. This is our elongation step. We have bound here and then red in this direction, and this yellow is our product. It's a single-stranded mRNA transcript made as a complementary copy to our template strand. So our mRNA strand, the yellow that it's making on this one, is going to be complementary to our DNA template strand. It's going to be analogous to the coding strand. All right, so we're making our mRNA transcript. And once we get to a termination sequence, we will be terminating. That's pretty straightforward. I don't think I need to explain that much further, right? Once we hit a terminating sequence, we'll stop making our mRNA and we'll just disengage, and the mRNA goes on and moves out of the nucleus and into the endoplasmic reticulum. Now, if you're in a bacteria, are we moving out of the nucleus and into an endoplasmic reticulum? No, because that stuff doesn't exist in them. So this can all just be right all in the same place in the cytoplasm. All right, here's a video of transcription.
File your taxes 100% free.
File your taxes, y'all.
Last year.
Do it earlier rather than later, and you know I'm right about this.
Yeah.
Do it earlier rather than later. My husband and I are talking about this right now.
And file free. That proteins are contained
Okay,
in
here we
our
go.
DNA. DNA contains genes.
Sorry, it just
A
reminded me.
gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. The motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete. and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns.
We're going to talk about this in a minute.
For the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail.
Don't worry about that.
added.
We're not going to talk about that.
This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the message.
Okay, so we'll come back to that. All right. So sticking with our transcription and into our translation. So our transcription going from DNA into their messenger RNA. That's all that is. Making a single-stranded mRNA copy of it. All right. Now, our translation is the whole point of the game, to make protein. That's what we want. The mRNA is not the goal. The protein is the goal. So how we're going to be doing that is reading our codon on our mRNA nucleotides, and that determines which amino acid we're going to add in which order. Okay. And that was what that talk was talking to us about, was our different codons that you can have. We have 64 different codons. We have about 20 different amino acids in most biology that you can code for with that. And then we have some that are stop codons, UAA, UAG, and UGA. Again, I won't test you on those, but there are ones that code for stopping. Basically, the fact that they don't code for an amino acid means stop. So we call these guys also not just stop, but nonsense codons. So nonsense codons means that they don't code for an amino acid. That's what it means. And that's going to become important in a little bit when we talk about mutations. All right. So if your genetic code on your mRNA said CCC, so if your mRNA strand said CCC, your tRNA anticodon would be GGG, and that would mean that this is attached to proline, amino acid. Starting to see how that makes sense a little bit? Okay, so we're going to see it. Your DNA triplet. This is your template strand on the bottom this time. This is your mRNA codon. This is the matching anticodon for that one. And then that would be attached to methionine. Okay, so this would be your template strand, your complementary codon, your anticodon codes for leucine. And then we will just hook these guys together as we go. And that's how all of this is going to work together. So again, this process right here, transcription. This process right here, translation. All right, translation also has 3 steps. Guess what they're called? The same. Initiation, elongation, and termination. Okay, we're always going to initiate at our AUG, which is our start. We're just going to elongate for until we get to a termination step, which is always going to be a stop codon of some kind. Which again, we can also call that a nonsense codon. All right, you ready to see how it works? Well, we already said this part. Translocation is the term for shifting our ribosome down as we're reading it. All right, so here's some pictures, static pictures of it. You have one guy coming to this first spot where your anticodon matches up with your codon. It's carrying methionine because AUG matched up with UAC. All right, next step, CUG matched up with the anticodon here. It's carrying leucine. We're gonna match these guys together by a peptide bond. Once that peptide bond happens, we're gonna get rid of our empty from our first spot and then shift that whole second spot guy over. And then see how we have our amino acids linked together by our peptide bond here. The next guy can now come in, match up our CCG with our GGC on our anticodon. These guys are matched up. We'll make another peptide bond and so on and so forth. Hopefully they have a video of this. Yes, thank God. Okay. This is way easier to understand if you see it in action. Come on, man, kick into gear.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor.
Don't worry about
The
that.
process is broken into 3 stages:
I want you to keep the basics, just the basics.
elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
All right, is that a little clearer for you guys? I like the videos better on these talks. I just can't draw it or explain it the way that the videos do. It's easier with the video. All right. Okay, so in bacteria, while you are making your transcript from your DNA, this is your messenger RNA, by the way, this yellow is, while you're making it, you can attach your ribosomes and start cranking out protein at the same time because you don't need to get it out into the endoplasmic reticulum. There is no endoplasmic reticulum. You're already in the cytoplasm. So you can literally start translation co-transcriptionally. So this is, again, what we call co-transcriptional. So during transcription, you can start translation. You don't need to finish your mRNA. You can just start translating it while you're transcribing it. We cannot do that in our cells. Why? because we have to move our mRNA out of our nucleus and into the cytoplasm, into the endoplasmic reticulum where our ribosomes are. We have to move the whole finished thing out. So these guys don't have to wait. So that's makes it a lot faster for bacteria. So this brings us to our concept of COVID-19 mRNA vaccines. So in our vaccines from Moderna and Pfizer, these are basically just mRNA that we put into a vesicle, just a vesicle, and that will allow us to inject it into your arm. Your cells will take up that mRNA and then just start translating it. And the protein that it's translating is going to be the COVID spike protein. And so now your cells are putting COVID spike protein all over their surface through this normal translation process. And then your body is reacting to that and having a reaction. That's why your arm turned red when you got your COVID vaccine and all of that, because your body was reacting to foreign proteins on the surfaces of your cells and eliciting antibody response against that. And now you have some protection against COVID infection, right? So that's how Pfizer and Moderna ones work. All right, we already talked about co-transcriptional translation. We mentioned briefly in one of those videos, introns and exons, remember? Exons are our coding regions. They actually have the information we need to make our protein. Introns are waste. They're waste DNA. They don't code for anything. So we have to cut out our introns and splice together our exons. This is only in eukaryotic genes. This is a problem, because that means if I put your direct genetic sequence from your DNA into bacteria that codes for insulin, let's say, the bacteria don't know what to do with your introns and exons. They can't make insulin from just your DNA. So what we have to do is collect your already spliced, ready to go mRNA transcript, this fully processed one, this heavily processed, fully ready to read by a ribosome product. We have to collect that and then turn that back into DNA so that the bacteria can read it. Does that make sense to you guys? Because otherwise they don't know what to do with introns. And so it took us discovering HIV to be able to engineer this, because HIV taught us how to go back from RNA into DNA. We didn't know how to do that in the past. We had to build it basically from scratch. And so now we can do things like this a lot more easily thanks to things like HIV. So a blessing and a curse, right? But all right, so let's talk about operons. These are the worst. Everybody gets confused. Operons are sets of genes only in bacteria. So only bacteria have these. Bacteria and archaea, but who cares about archaea, right? This is supposed to be bacteria and archaea, but okay. So these are sets of genes that are regulated all together. And expressed all together as one unit. So a whole set of genes that we're going to control all together. So these would be genes that might be working together to make a nutrient, or genes that are working together to break down a nutrient, stuff like that. Okay, so we have inducible operons and repressible operons. We're going to talk about inducible first. Inducible. If you're going to induce labor in a person, so a lot of people relate inducing into, we all know what inducing labor is. If you have to induce labor in somebody, were they already in labor? No, right? Otherwise you wouldn't be inducing it. So please remember that, okay? If we are repressing something, that means it was already being expressed and we're trying to repress it. Okay, so those are the two differences. So inducible operons, these guys are off by nature. Wow, I just cannot write today. Off by nature. And we can turn them on, okay? Repressible operons are on, and we can turn them off. All right, so we're going to start with examples. The lactose or the lac operon. Okay. Basically, you will have the promoter, which you guys remember, this is in DNA. You'll have your promoter region. Let's do this. Promoter, we'll put a P for promoter. This is where your RNA polymerase binds. And then your gene would be all after that we would be making our mRNA from. Okay, we understanding that? Okay. All right, so we have an operator for these guys that'll be upstream of that. Or downstream, it doesn't really matter. So let's actually put downstream because I feel like that makes more sense in the picture. I want it to be confusing. So we'll put our operator down here. And then there's our product, okay? What happens is this guy has a big old fat protein that sits here and blocks RNA polymerase from moving down and making the product. So at our operator, we have this big old fat blocking guy. And this big old fat blocking guy is called a repressor protein. It doesn't matter if we're talking about inducible or repressible. Both of them have repressors, okay? So in our lactose, the reason that it is off and needs to be induced, so this is our inducible example, is because the repressor is always on there, unnaturally blocking. So we're turned off. Our gene expression is turned off. We can't make anything downstream from here because we have a repressor sitting there normally. So what happens with this is that what we will get is lactose, which will be this green guy here, okay? Which is just sugar, lactose. You guys know what lactose is, it's in your milk. So lactose will come and bind onto this repressor and force it to jump off. And then the polymerase can go through, okay? So when lactose binds that repressor, the repressor comes off and stops blocking, and now we can make our mRNA. So we'll only turn on the genes that break down lactose when lactose is present. And so that's how we'll control them. So these genes that are downstream here, mRNA of genes to break down lactose. Okay. So we'll only turn on making those genes if lactose is present. Why would we be making that if lactose wasn't there? Why would we be just making that all the time? Because otherwise, if we don't have this system in place, you're making this all the time. What a waste of your resources. So we'll only turn on making these genes when lactose is present. The way that works is lactose binds the repressor comes off, and now our polymerase will start to go. And that's how we induce that, okay? All right. So our repressor protein basically is allosteric, meaning that there's a part of it that'll bind with lactose and cause it to dissociate. So we can transcribe. Here's a better drawing of it. Normal situation, operator is locked by a repressor protein. Lactose inducer binds onto it. It comes off. It's hanging out here inactive so that our polymerase can come in and transcribe. We transcribe, and that leads to translation of these genes. That'll then break down lactose. Once we've used up all the lactose and the lactose is no longer associated with that repressor, it'll just happen naturally. Once we use it all up, then we'll turn this off because we don't need to keep making it anymore. And that's how it's all controlled. It's the lactose. That's only one of them because now we have a repressible operon. That's inducible. Now we're going to talk about repressible operons. These will only be turned off whenever we don't need what it's making. Same idea. These are always on. Can be turned off when we don't need it. Obviously, if you have lactose only sometimes in your diet, you only want to make the genes to break down lactose when it's there. So you'll turn those genes on only when lactose is there. So that's inducible. This guy is basically making amino acids or other macromolecules. We're making macromolecules all the time, but sometimes you have an excess of them and you don't need to keep making them. So we'll turn them off. That's what a repressible operon will do for us. Don't need to keep making these macromolecules. Stop making the macromolecules. That's what our repressor is going to be doing for us here. Okay, so we have normal situation. This is how we're going to start. We'll be transcribing down here. We start making our product. In this case, it's arginine, which is an amino acid. So all of our genes that are involved are making arginine. Normally, your body will use that up and it'll go away on its own. But if you start having an excess of arginine, so we have excess product, people aren't buying your jeans. So let's say like actual pants. People aren't buying your pants or your dresses or whatever it is. Okay, you're at an assembly line, you're at a factory, and people aren't as like your chinos or whatever that you're making are not as popular anymore. So why are you going to keep making them? You're going to make less, right? So that's what this is all about. People are not ordering your chinos anymore, so you don't need to keep making them at your factory. You have an accumulation of excess of product there. So this excess product, this binds with our repressor, and now it binds with our operator and turns it off. So now we're blocking the exact same way as the other one. So you have how these are literally set up the opposites of one another. So remember that the inducible operons, you are off and you'll induce them to be on. The repressible operons, they are on and you'll induce them to turn off. Make sense? Okay, there's video on it. Real quick. Hopefully this one's not too long because we don't have a lot of time.
Operon is a group of genes that transcribe a single mRNA. The structure of an operon can be remembered using the acronym PROG, which stands for promoter, repressor, operator, and the genes. At the very end of this structure, there is a termination sequence which terminates transcription. Usually, upstream from the operon is a regulatory gene encoding for a repressor which can bind to the operator. RNA polymerase is responsible for transcribing the genes of the operon. So let us take a closer look at these individual parts. First, the promoter is where the RNA polymerase binds to initiate transcription of the genes. The operator can be thought of as a stoplight that either allows or prevents transcription of the genes by RNA polymerase. This depends on if the repressor is bound to the operator or not. If the repressor is bound to the operator, it prevents the RNA polymerase from transcribing the genes. If, on the other hand, the repressor is not bound to the operator, it allows the RNA polymerase to transcribe the genes. If the repressor is naturally bound to the operator, it is said to be repressible, as it requires external changes to stop binding to the operator. If the repressor naturally remains unbound, it is said to be inducible, as it requires external changes to start binding to the operator. An example of a repressible operon is the lac operon, and an example.
This is opposite explanation of what our textbook gives. So this actually is really frustrating for me because I did not rewatch this. But but that gives you the gist of the operons and how they work. Remember, lac operon, it will be a whole operon is induced. The whole operon is induced. Okay. Anyways, all right. So DNA recombination events. Basically, a recombinant is any organism that's going to have any genes from any other organism. So anytime you have that happening, that's going to be recombination. You can transfer genes from one organism to another organism without it being from your mom or your dad. We don't really do that too much here as eukaryotes, but bacteria do it all the time. They can shuttle genes through their channels next to each other through those sex pili. They can literally make a copy of their plasmid and just shuttle that plasmid through that little channel, that pili, through that process called conjugation. Plasmids are really easy to do that with because they're small. They'll have extra genes that help in survival. They're not necessary for survival. They just help it be even better. Okay. Again, that's called conjugation when we are sharing that. Transformation is another form of horizontal gene transfer where you're just picking up foreign DNA from outside of the cell. Not all cells can do this. If you are able to pick up genetic material from outside of your cell, you are called a competent cell. We'll talk about this in lab 10, I think it's 10. And then transduction, this is where we got into that idea of how your phages will incorporate their DNA into the bacterial genome, the prophage situation. That process is called transduction. Okay, so transferring genes by phage. mutation is anytime we have a change to our nucleotide sequence. A lot of times these are spontaneous. We'll talk about some of the mechanisms by which that can be happen. So this is spontaneous, it's just be happening because DNA polymerase is not perfect and will lead to random mutations just from imperfection. We also have induced mutations. This is due to exposure to things that are known to cause errors in the genetic code. So this could be things that we know of as carcinogens, or by exposure to like UV radiation, it causes your nucleotides to link next to each other instead of bond to the guy across from you where you're supposed to be binding. And then that causes misreading of that DNA, cause damage to it that way. So the different kinds of mutations we can have: missense, nonsense, silent, back. Okay, so missense, we are going from one amino acid to a different amino acid as a result. Nonsense, we're going from an amino acid to a stop. So we're either changing your original amino acid to a different amino acid in a missense, to a stop codon in a nonsense, or a silent mutation. It's the same amino acid, but if we looked at that chart before, way back when, oh my gosh, now I'm finding it. It's going to take me forever, isn't it? Oh my gosh. Okay, I should have just done this. Did I close it? No, I didn't. Okay. This chart here. God damn, here it is. This. So leucine is actually coded for by AUU, AUC, and AUA, for example. So if you change from AUU to AUC, you're still isoleucine. You're still coding for isoleucine. You get what I'm saying? So you can still be coded for by multiple amino acids. That's a silent amino acid change. Okay. And then a back mutation is going back to what the original would have read. Okay. All right. We're almost. Then a frame shift mutation. Imagine that you are reading a book that says the cat in the hat or whatever, and you take out one of the letters, but you shift it back over. So now it says the cat, it, he, at, whatever. Suddenly now, if you keep on reading the whole book that's been shifted by that one letter, suddenly now that whole book doesn't make any sense at all. And so that is the same idea with frame shift mutation. You take out or delete one or two amino acids, and now that codon reading frame has been shifted in such a way that you're no longer making the amino acids downstream that you would have been. So these can be disastrous and even deadly as a result of that. So this is image that you guys can refer to that'll show you the difference between those from the original sequence being changed. So this will be your normal. You can look at the different ones and the results of them as they go through. So if you have an early stop, suddenly you're not making those products. And then everything downstream can get affected by a frame shift change. All right, and then we have a test called the Ames test that we will use to take salmonella that can't make an essential amino acid for themselves, expose them to something that we think could cause mutations. And if that mutation makes it so they can now make histidine for themselves, we already changed them so they shouldn't be making it. We should have to provide it to them. And now suddenly if they don't have to have it provided, that means they had a mutation that caused that to happen. Okay, so that's called the Ames test, where we use bacteria to determine the ability of things to cause changes in your DNA, their carcinogenic potential. And a carcinogen is something that can cause cancer because if it can cause changes in your DNA, it can cause changes in the genes that control basically your cell replication cycle. And now you're replicating out of control. So that would be a problem. All right. So our questions: explain what makes DNA semi-conservative. DNA replication is semi-conservative because whenever you go from old strands to the new daughter strands, each daughter is going to be part old and part new. Okay, part old and part new. During DNA replication, which strand makes Okazaki fragments? That is the lagging strand. Great. What is a codon? It is a set of 3 nucleotides on the mRNA specifically. It codes for the amino acid when we're doing translation. What type of operon is usually on and will be turned off when enough product is made? The operons that are on and can be turned off will be called repressible. The operons that are doing that. If a mutation causes AAA, which codes for lysine, to change to ACA, which now codes for threonine, and you see all we did was change the second one here. but still different amino acids. What type of mutation is that? Missense. Different amino acids, all right? So that's that. Let's go ahead and head up to the lab.